So before we get any further, what I would like to do is to go over just one of the basic concepts concerning how we classify organisms into taxonomic groupings, or taxa. And I need to get you to the point where we can talk about a key idea that's going to be indispensable for our discussions, and this is the concept of a synapomorphy. I'll be adding this 5-syllable word to your vocabulary today, but a little bit of background is going to be necessary before we get there. So let's take a little bit of a break from our discussion of plants and talk about the process of taxonomy and the whole idea that we have these things called taxonomic groups. So a couple of words for you are taxon / taxa, and the plural of taxon is taxa. So they're really only the same word. It's just one word. And because we're living in the 21st century and we can forget all about the sloppy ways that this term has been used in the past, I'll tell you right here. When I say taxon, what I mean is a grouping of organisms united by common ancestry. And I really should clarify that this is a definition of a good taxon, because as I implied before, it's also possible for us to use taxon in a very incorrect way. And so maybe the best thing for me to do here is to illustrate with a grouping that some of you may be familiar with. Okay, you are going to be familiar with this. We'll be talking about the Felidae, or the cat family. And so Felidae is the official Latin and universally valid word meaning cat family. If you have the -ed at the end, we're talking about the family level of rank in the taxonomic hierarchy. And so this cat family is going to include things like lions and house cats and cheetahs and pumas and ocelots and tigers and all the other ones that you can think of. And so you're probably saying, yeah, this is simple because quite obviously they're closely related animals, and you might point out that they've got a lot of similar characteristics, and you'd be perfectly on the right track. But what I'm saying here is that the only valid reason why it's legitimate to put house cats and cheetahs and tigers and pumas all into the same family is that they share a singular common ancestor that they do not share with the rest of the non-cat animals. This thing right there is our common ancestor to all cats. And we suspect that this animal lived in the distant past, maybe 55 million years ago. 55 million years ago, we didn't have different kinds of cats. There was just one cat. And yeah, it was related to dogs and everybody else, but this one cat gave rise to the entirety of the cat family, the entirety of the Felidae. And this is a true story. This is a real and true story that all living cats and all extinct cats can trace their ancestry back to this one ancestral species. And what I'm telling you here is that this is what makes the Felidae a biologically valid taxon. Now, you could take a wolf, and you would say that the wolf is obviously not a part of the cat family because you might argue that it lacks cat-like characteristics. But it might be conceivable that you might have some type of dog or maybe a weasel. Okay, so maybe you got this weasel and it's a cat mimic. I call it a cat mimic weasel. And so even though the weasel kind of looks like a cat, it might have evolved some cat-like characteristics. This would still not qualify this animal to be a biological cat. And the reason for this is that this animal, wolf or the cat mimic weasel, is not part of that network of animals, the Felidae, that all trace their ancestry back to this common ancestor. The wolf and the cat-like weasel would each have its own family tracing backwards through their own ancestral histories, but neither of those ancestral histories was going to include this animal right here, and therefore they don't belong in the cat family. Pretty easy? Good. So now that we've gone through the theoretical underpinnings of this absolute rule of modern taxonomy, that a taxon is valid if it's defined as the grouping inclusive of all the descendants from a common ancestor, I'm going to reintroduce the term synapomorphy, which is going to be crucial as we move forward with our discussions on plant evolution. It's a completely practical element of taxonomy, and despite the fact that the word is long, it's actually a pretty easy concept. You probably already have this idea that the lions and the house cats and the cheetahs and the pumas are all members of the cat family that are united by hallmark characteristics. Maybe you're looking at some feature of the cat's anatomy. Okay, there's a really good cat. Maybe the whiskers, maybe the ears, maybe the tail, maybe the incredible flexibility of cats. This might be your working list of the characteristics that define cats. Okay, so a synapomorphy is basically this. A hallmark characteristic that both defines and is limited to a particular taxon which is linked through a common ancestor. So maybe your working list of synapomorphies for the cat family might include a really sinewy tail, might include these whiskers, might include the flexibility, and that would be potentially a working list for you. Now, if you were an expert in mammal biology, you might come up with a different list that might be a little bit more rigorously defined. So let's do this. Let's ask a mammologist. Hey mammologist, what are the synapomorphies of the cat family? And they would come back with a list that looks like this. And so a lot of these characteristics are dental characteristics. M1 is the first molar. And we're saying that the first molar is reduced to a small transverse bar, and it lacks a talonid and a metaconid, whatever those are, right? And so, according to the experts, these are the synapomorphies of the felidae. Now, if you had this list, and then I showed you an animal that you've never seen before, you'd be able to tell pretty quickly whether or not it was a cat, based on whether or not it fit the description required by this list of key characteristics. Maybe it's a new species being described for the first time. Maybe it's a fossil species that's never been looked at before. You'd be able to decide whether or not it's appropriate to assign this new species to the cat family. So this is a pretty easy idea, right? But what does it really mean? What's the reason for there to exist such a list of characteristics? We're going to start calling them synapomorphies. We've got a list of synapomorphies that are unique to cats. Why do all the cats have them, and why do all the non-cats lack them? Well, if you think about it for more than a couple of seconds, it should just make perfect sense to you. All of the characteristics on that list were present in this ancestral cat species, right? This animal here probably had a lot of other characteristics that were not passed on to all of its evolutionary descendants. Those characteristics that were passed on to all the descendants are basically the synapomorphies, the things that were inherited unchanged from that common ancestor. The reason why these guys are linked together is because they share this common ancestor. The reason why they have these common characteristics is because they inherited those characteristics from the ancestral cat. Now, a cat like weasel might have certain cat-like characteristics, but it didn't get those characteristics from this ancestral cat. It could not have, because this is not an ancestor to the cat-like weasel. Okay, so now that I've inserted this humongous word, synapomorphy, into your vocabulary, let's try using it in a sentence. And let's make the sentence meaningful for our understanding of the evolution of plants from the Ordovician period. Boom. I'll read it out loud for you. Many of the key synapomorphies of land plants are functionally related to their ability to succeed under the harsher conditions presented by life on dry land. First, you have to remember that plants, and I mean the taxon consisting of the whole plant kingdom, are defined by their history of having crawled up onto dry land during the Ordovician. What we're saying here is that we've got these characteristics that are unique to land plants. Basically, you find them in all plants, but you don't find them in the non-plants like the algae. And the way the sentence calls them synapomorphies, it's saying exactly that. We're also saying that these key characteristics of plants, their synapomorphies, are directly related to their ability to survive in their new dry land habitat. Remember how we talked in the previous video about how the Ordovician, and especially the end of the Ordovician, is associated with a drop in sea level and a cooling and drying of the Earth's climate. Well, it should make perfect sense that during this period, the earliest plants were under intense pressure to persist in the face of dry periods, times during which the plants would need to withstand drying out. And again, if you read the sentence again, you'll see that this is exactly what was said. Okay, so I've told you that the Ordovician was a time in the Earth's history in which we have the origin of land plants. Some kind of alga-like protoplant finally crawled up onto shore and became the first plant. But fossil evidence that could tell us exactly what kind of plants these were is entirely absent. Plants in general don't fossilize as well as, say, clams. Okay? If plants have any hard parts at all, they just aren't as durable as clamshells. And most of the parts of most plants are just too flimsy to have much of a chance of being fossilized at all, except under the most unusual of conditions. But one part of these drought-resistant early plants that do fossilize reasonably well are the spores, which are the stage in the whole life cycle that sticks around during the dry times, growing into a plant only when the conditions become wet enough. So in the case of these Ordovician land plants, we have a few spores, and that's about it. Nothing else to tell us what kind of plants the spores grew into, just the spores. But we can tell that these spores are from land plants and not from algae because of some key characteristics. What was the term again? Yeah, synapomorphies. Land plants use a unique material called sporopollenin to protect the spores and pollen grains from drying out. The other synapomorphy is a trilete marking pattern that you get in the individual spores, coming from the way they split apart from each other. So spores are always produced by this process called meiosis, where you have one cell that will eventually split into four, but the four cells that come apart are going to be packed into this ball-shaped structure. Okay? Basically, if you take this tetrahedral packet of spores and you pack them into a ball, you get what is going to be the structure that will eventually cleave off four individual spores. Okay? And when one piece cleaves off, what you're going to see is this trilete pattern on each spore. It's going to look like this, right? Kind of like a Benz logo. And the trilete spore characteristic is a synapomorphy of the plant kingdom. All land plants have trilete spore markings, and no algae or no other non-plant has them. And because we have from the Ordovician trilete spores that have sporopollenin, this is enough evidence to unequivocally support the existence of land plants at the time when those spores were deposited. Again, that was the Ordovician. We have to presume that the reason we don't get the other parts of the plants fossilizing is that they were soft, like what you would find in many of the non-vascular plants today, the mosses and the liverworts and the hornworts. When you don't have reinforced, thick-walled cell structures that transport water or support tall plant structures, there won't be anything except for the spores that are durable enough to fossilize. Now, during the Silurian, we start to see some of those more durable structures in the earliest vascular plants, like Cooksonia, which grew to the towering height of about 6.5 centimeters. So the story we have from the fossils is that the earliest plants existed in the Ordovician. We've got the trilete spores and the sporopollenin. And since we have good fossils from the Silurian, like Cooksonia, we've got evidence that vascular tissue exists at that point. Now the presumption here goes that the earliest plants of the Ordovician lacked vascular tissue, similar to modern mosses and liverworts, which are also non-vascular. What this suggests is that the earliest land plants from the Ordovician were non-vascular, and the earliest vascular plants were things like Cooksonia that may have evolved as early as the latest part of the Ordovician, but more likely sometime in the early Silurian. We can also tell that the state of being vascular is obviously not a synapomorphy of plants as a whole. We don't see it in the mosses and liverworts, and we don't have any evidence of it from the earliest plant fossils. The evidence I've presented so far tells the story of early events in the evolution of plants. Specifically, we're looking at the mosses and the hornworts and the liverworts today, with their lack of vascular tissue, and concluding that they must have split away from the rest of the plants before the evolution of vascular tissue, which we already see in Cooksonia. This point at which plants first had vascular tissue was the starting point for the evolution of Cooksonia, as well as all the later plants that were vascular and eventually grew bigger and better as land plants. But these non-vascular plants over here, they don't have this vascular plant ancestor in their history. Now I'm going to go into a little bit more detail here. We've actually got 4 different lineages: the Marcantiophyta, or the liverworts, the Bryophyta, the mosses, the Anthosrhodophyta, or the hornworts, and the rest of the plants. And I'll put a name on them later. So let's use synapomorphies to decide what's the most likely history for the splitting of these 4 plant lineages. Who split off first, who split off second, and who split last? So way back at this original plant ancestor, you're going to have one group splitting away, and it's going to be the most basal, or closest to the bottom of the tree. Usually, this is going to be the one with the most primitive characteristics. And in this case, it's the liverworts, or the Marcantiophyta, that fits the bill. Unlike the mosses and the hornworts and the rest of the plants. Liverwort sporophytes lack stomata, the little holes through which the plant's leaves take in carbon dioxide and release oxygen. So the two branches splitting away right here is going to be one leading to the Marcantiophyta, and this other one is going to evolve stomata. From this ancestor, all the descendants are going to have stomata, and that's going to be a characteristic of that taxon. So this stomata-containing common ancestor there, again, is going to split into two different directions. One of which is going to give rise to a more basal group, and the other one is going to be more derived. Okay? In this case, we think that the bryophyta, or the mosses, are going to be the more primitive group because they lack a characteristic called indeterminate growth, or extended growth of the sporophytes. Bryophytes have sporophytes that grow up to maturity and then they stop growing. It's determinate. Whereas with the hornworts and with everybody else, the sporophytes continue to grow even after they produce the sporangia. The final split is going to be between this lineage, the one that gives rise to the Anthoceratophyta, and this other one that's going to give rise to the Cooksonia and the rest of the vascular plants. Now the way I've drawn this, the key characteristic that separates these two guys is the presence or absence of multiple sporangia on the sporophytes of the plants. It turns out that Anthoceratophyta, like the bryophytes and liverworts, they only produce one sporangium per sporophyte, whereas in the case of Cooksonia and the rest of the vascular plants, we have multiple sporangia on every sporophyte, and therefore this is the important characteristic that unites all plants in this grouping. In fact, we're going to be using a different term to represent all plants in this taxon. Okay? We want to replace the endpoint here with a different term, and that term is going to be the polysporangiophyta. That basically means everything else in the plant kingdom except for the liverworts, the mosses, and the hornworts. Now this particular tree is making some very specific predictions about what we think was the sequence of splittings in the earliest days of the plant kingdom. We think the liverworts split off the earliest, then the mosses, and then the hornworts basically split away last before we had the evolution of the polysporangiophyta. This tree is based on the key characteristics, or synapomorphies, that can be observed in the physical structures of the plants themselves. Now we're going to look at a reconstruction based on the molecular sequences from these organisms. You can take the DNA from the chloroplast and compare the sequences from several mosses, hornworts, liverworts, and polysporangiophytes. And when we do this, we find that the mosses all tend to be similar in sequence to the other mosses, the liverworts similar to other liverworts, and the polysporangiophytes similar to other polysporangiophytes. And so far, this is exactly what you expect. We also found that the hornworts were most similar to the polysporangiophytes, and vice versa. Polysporangiophytes are similar to each other, of course, but the next closest similarities were to the sequences of the hornworts. It also seemed like liverworts were the most dissimilar to all the other plants in our sample. So what do you think? Are the DNA findings that I just described consistent with the layout of the tree that I'm showing you here? I hope you said yes. Molecular data and morphological data are not always in agreement, but in this case, which is actually pretty consistent with what we find in real molecular studies, they do agree. And we can take from this consensus of two separate analyses telling us the same thing, that this reconstruction, this particular reconstruction of the evolutionary history of these plants is pretty likely to be the truth. Here's another question for you. Look at this tree and tell me, is a moss more closely related to a liverwort, or is a moss more closely related to a pine tree, which is in the polysporangiophyta? Now, if you said the moss was more closely related to the liverwort, you're probably just looking at their positions relative to each other on the tree the way I drew it. But look at this node right there, right? It's completely arbitrary whether I draw the bryophyte lineage going up or I draw it going downward. I could easily have shown you a tree that kind of looks like this with the Marcantiophyta over here, and the bryophyta are pointing in the downward direction, and the anthoceratophyta are going to be more basal, and the polysporangiophyta are going to be out here at the tip. This second tree I drew here is exactly the same as a tree that I drew above, only this time mosses and liverworts look like they're on opposite sides of the tree. Now, the only thing that matters in deciding whether a pair of organisms is more closely or more distantly related to each other is the point in the time in the past in which they shared their common ancestry. Mosses do share a common ancestor with both liverworts and polysporangiophytes, but these two ancestors are not the same. Okay, so let's say this. Look at this. We'll name these ancestors. We'll call this Ancestor A. Ancestor A is the most recent common ancestor shared by the Marcantiophyta and the Bryophyta. The most recent common ancestor shared by mosses and polysporangiophytes is not A. It's this guy over here. We'll call him B, him or her B. Okay. Ancestor B, the one shared with polysporangiophytes, existed more recently in the past compared to Ancestor A, the one that mosses share with the liverworts. And for this reason, the mosses and the polysporangiophytes have to be more closely related to each other than mosses are with the liverworts. Now remember, polysporangiophytes is a large grouping including all plants that you're familiar with, like redwood trees and orchids and pine trees. This actually does mean that a moss is more closely related to a pine tree than it is to a liverwort. This is going to be surprising to a lot of people. Mosses and liverworts do share a lot of primitive characteristics, but this does not mean that they're closely related. This is a reasonable point to break before the next video. To summarize, we started off with a very basic introduction to the way that we like to organize life into good taxa that are natural groupings of organisms related through common ancestry. Under most circumstances, we can identify group-wide commonalities that are also unique to the group and are there because they are inherited from the common ancestral species that gave rise to the group. These are the synapomorphies, and this is an important concept for us. We use synapomorphies to recognize and identify organisms as belonging to their appropriate taxa, and the one place where this is especially important is when we use fossil life forms to reconstruct the history of life. Finding highly incomplete fragments that bear the synapomorphies of plants from the Ordovician, that was enough to say that the plant kingdom originated then, even though we don't have any good plant fossils until later in the Silurian, when the plants had become more vascular and sturdy-celled. We also ran through the earliest history of plants, leading to 4 different kinds of plants: liverworts, mosses, the hornworts, and the polysporangiophytes. So we're going to look at the subsequent evolution of this last group in the next video.